So with that, so the two people that will present is from the EU side is Simone Baconnier, and then Scott McNeil, who is a director of NCL, and he will take questions. Scott, you need to take questions at the end.
So we agreed. Thank you very much, Anil. We agreed with Scott that I'm speaking and he's answering the questions. Okay. So that's a perfect duo. Well, thank you very much for joining today. I have the pleasure to present one of the most concrete and most successful outcomes from this nanomedicine US and EU collaboration, the EUNCL raised, birth, and let's say first year of existence. So we are still a very young infrastructure compared to the 12 year of advance the NCI-NCL has. And I'm just going to show you how this huge experience the USNCL has and how they brought that to us in order for us to avoid some mistakes, even if as a young existence we have made some mistakes anyway, and how we optimized our growth thanks to this support. So, to make the story short, those three musketeers are really at the basics of this, let's say, setting up of the EUNCL and the transfer of the knowledge from the US to the EU. So Scott from the NCI-NCL, Anil from the FDA, but formerly from the NCI-NCL, and Patrick. There were, let's say, historical discussion before UNCL could start, but not so much long time ago. But anyway, those really three people are the musketeers, and we really thank you for providing your support. So EUNCL is what we call in the technocratic EU language an infrastructure. We have been funded by the European Commission to start to work in 2015, and we have a 4-year program with 3 application campaign, which we are ending this year. And the main objective were really to, let's say, build up a mirror institution or a mirror infrastructure mirroring the US NCI-NCL. So we are, let's say, a distributed infrastructure among Europe plus the US, which is really as well a very good example of the capacities of EU project to integrate US team. It's quite rare, but this is a successful example where we have all our European labs and the US supports in the setting up and in the running of the facility. So as you can see, we have, let's say, different capacities, whether they're in vitro, physical, chemical characterization, all over Europe, with a really strong support of the USNCL from the beginning, from the really start up to now on a weekly basis. And I'm going to show you where this support was really important. So as you all know, Europe has some difficulties in terms of organization. We are spread all over, let's say, different countries, cultures and things like this, even regulations. And it's quite often that it's really hard to work together, or it's not easy to work together. And really here, the multiple, let's say, non-integrated providers we have gathered in UNCL among the integrated multidisciplinary infrastructure is really as well a key success in Europe, and EU funding are really welcome for doing this kind of let's say, to build these kind of initiatives. So we have now in UNCL a robust, I forgot an N, a robust, standardized and regulatory relevant assays, a set of assays. And I'm going to show you how we reach this level of, let's say, quality level thanks to our US cousins. So we went through a process of quality development, which was organized around SOP transfer, bootcamp and training, cross-validation, and finally kind of a roadshow around bugged samples, which were challenging, and we fortunately managed to let's say, go through all those challenges with success. So first, SOPs, definition, transfer, and cross-validation of these SOPs took quite a while. The USNCL, let's say, provided us with a list of SOPs on different techniques to bring, let's say, sets of data to our sponsors or client, whatever you call them, in order to really support the development of this dossier with key characterization and characteristics. So we benefited here from these SOPs, let's say US version, that we transfer, modified when relevant, and let's say qualified in our institution, in the different labs are presented in the network. And we benefited here from several years of expertise and more than 300 case study of the NCI-NCL. This was really strong experience sharing, and we have also, of course, adapted but also produced new and additional SOPs related to the EU environment or to the EUNCL capacities. So the second, let's say, level of cooperation, which was also a very concrete example of collaboration, we have made a bootcamp in the NCI-NCL for 2 weeks. So we have had 13 of our EUNCL core expert team members who went in during December 2015, which is, let's say, around 6 months after the beginning of the project, for a real concrete technical training and operational process and management training. So we have had really fruitful 15 days of work. Very much thank you to the NCI-NCL because they had to stop their activity for 15 days because of safety and, I'm missing my words, and let's say IP issues. So really thank you. It was really a strong and very, let's say, a team-building adventure as well. Third step, we have had a cross-validation plus a qualification. So we have qualified all our SOPs, the one we transfer from the US to EU, against the SOPs. So we have worked on the research-grade liposomal doxorubicin solution. All the labs have carried their the assays on this specific batch product to really see if the results we could propose using these SOPs were, let's say, in the range of usage. And we have had also a method cross-validation on a specific compound between the US and the EU using the same cascade, the same assays, and we reviewed our results with success. So results and reports were reviewed by both. We crossed them and compared successfully for US and EUNCL, sorry. So the last step we had in this cross cooperation and this support from the US in terms of optimizing and making really a success of the setting up of EUNCL is the bug sample campaign, where we have had a series of 4 samples which had different bugs. Like for the endotoxin, we had a contamination we should have recovered. The drug loading was a wrong drug. We were informed about a drug, but it was not the right one. The composition, aninity and purity issues, and additional compounds in a nano product. And the cytotox, where we discovered that it was not the nanoparticle who was toxic, but the supernatant. So we managed those challenges with success, hopefully, and it was really, again, a great, let's say, success for the setting up of EUNCL. Well, EUNCL intends, and unfortunately, it takes time to have a kind of a central role as the NCI-NCL has. Fortunately for the NCI-NCL, there is, let's say, there are agencies above the NCI-NCL, which are the FDA and the NCI, and this direct link with the FDA is really crucial. We're trying to set up the same link at the European level with the EMA, the European Medicine Agency. It's not easy, it takes time, because EMA is one agency, but we have 27 others in each of the country in Europe. 28, sorry. Maybe soon 27. Well, so it's a bit more complicated, but we're, let's say, in progress, and I think we will reach, let's say, not the level of interaction the NCI and the, let's say, the level of, collaboration we have, the NCI has with the FDA, but at least a really good level of interaction with the EMA and the national regulatory bodies. So we're still in progress on that, but we're still, let's say, benefiting from the NCI-NCL experience, and we're ongoing discussion with the NCI-NCL for improving this. So this is just an illustration of where EUNCL tries to stand beneath the researcher and developing medical nanoparticles and the future producer of these nanomedicines, as well as the scientific community and the regulators, as I earlier introduced. And I would like to thank you with this. And Scott, please. Come, yes. So I'll leave the floor to Scott McNeil for the conclusion and any questions. No conclusion. Or no word, no additional word? Yeah, word.
There we go. Conclusion is, just like you said, passed with A+ marks. It's been a privilege and a rewarding experience to see EUNCL start up. Really, it comes down to person-to-person interactions throughout. And as you start up other programs, I encourage you to keep that in mind. Again, it's rewarding is to see the success that you guys have had over the last, what has it been, 3 years now, compared to that first conversation many years ago. So well done. But happy to answer any questions. Can you identify yourself, please?
I'm Frank. I'm Frank, and that's just what I am. No, I work for FDA in the Office of Chief Scientist, in the Office of Commissioner, the Food and Drug Administration, USA. The development is truly great and impactful, even if some of the things are still in the works and people cannot easily measure and count peanuts or whatever people count. But I think it plays a greater role in terms of stimulating innovation. And many of you know that we have recognized that at the agency, and our interest to build communication platforms to innovate at the agency how we review product and how we learn about science and new technologies. For example, we have created centers of excellence in regulatory science and innovation with academic institutions. But this platform is an excellent example how we could, whether we call it for FDA a center of excellence, or if we support it in one or the other way is a different question. But I think having this in mind as a resource that directly supports the mission of the agency, I believe is a great opportunity. It's also a great opportunity in context of public knowledge management, because at FDA we struggle with things to communicate into the public that is public information. And I think that what you have learned on the basis of SOPs and standardization that becomes available to be developed into consensus standards through the standards organization is a very important pathway that we should model after also in other contexts. So I think the public resource and the drive for innovation of such an international collaboration, but also as a partner with regulatory agencies, is significant. And because it is a public platform in a sense, the relative neutrality, or let me say it in other words, to exclude preferential treatment or perception of conflicts of interest or conflict of interest is really great. So I'm looking forward to continue and build out this collaboration also with the agency, and fortunately we have McNeil representing the agency very strongly in that. But I think it's also a question of the community to recognize this opportunity, that this is one path to communicate about science, the problems, and solve problems collaboratively in regulatory science in conjunction with agency scientists.
Yeah,
Not a question, but a comment.
Yeah, well said, Frank. And what happened behind the scenes as we were setting up is we surveyed some of the needs and gaps that the regulatory agencies had to make sure that our structured approach was in line with those needs, those gaps. And so we share a very fruitful relationship with FDA in regulatory science, as well as discussing scientific problems. We even are undergoing a collaboration on drug release assays to go after some very difficult problems that some of the reviewers are having. So it works at multiple levels. Well said, Frank.
Another question, Robert?
I would be interested to know. This was, of course, from the beginning, a rather unequal relationship because you had all the experience and EUNCL was just starting. EUNCL has learned a lot from USNCL. During this process, have USNCL also benefited from this process?
Oh, certainly, and certainly the National Cancer Institute has as well. As Patrick said, cancer is no respecter of boundaries, of borders, of tariffs, or of tweets, is what I said the other day. So we benefit altruistically, but also we're a full partner in the Horizon 2020 program.
Other questions? Questions or comments, Frank?
No, it's this time a question. How do we manage knowledge? So knowledge management in the context also in terms of education, because that wealth of information that is accumulated with the practical experience of testing, we need to communicate. The SOPs is one form. Developing it into consensus standards is another very important format. But I think also in terms of let me call it academic, or historically always referred to as academic training and education. How do we build this bridge better? Because I think academic institutions are looking for this opportunity to gain knowledge, particularly practically applied knowledge for their educational programs. And I think that's what makes really better professionals as they enter into, let me call it, the professional market. The preparedness of scientists to enter the environment in which they will work.
Thank you, Scott. If I may. So regarding SOPs, they are publicly available both in the US and in Europe. And regarding the standards, we'll have this next presentation is addressing exactly what we have initiated for going, let's say, from these SOPs to standards, common standards. And regarding education, our thing, but I suppose you do the same. That means our team, the EUNCL team on European grounds, is attending, I don't know how many meetings, conferences per year, always to present these lessons learned workshops to in places like these. So we had this morning the session on EUNCL. And, but we realized that there was some more demand, I would say, from the academic side, as you pointed out. And so we are currently thinking about, let's say, how to develop, let's say, educational modules for applicants. That means people developing nanomedicine to first understand why should they perform characterization, what are the regulatory needs behind in terms of timing, and what are the market needs at the end. So because we do it at the moment, let's say, on an individual basis by providing some kind of counseling to the applicants. But I think, and you are right to point this out, we have to provide it at a larger scale. So this is in our to-do list for the second phase of EUNCL from 2020 onward.
Lisa?
I just want to follow up on that. I mean, one easy way to broaden the impact, aside from doing workshops embedded in events like this, is maybe this core could do a series of webinars. Yeah. We do that. We archive them. People see them, for years after we do them. But that might be low-hanging fruit as an activity for this group.
Thank you for the very good suggestion.
Okay, Bia, do you have a question?
No, I just asked myself when Scott said we are within Horizon 2020, what is the pendant to Horizon 2020 on the level of US? Is there a program dedicated to the development that could be seen in a way as a Horizon 2020?
So, Scott, do you know? Lisa? No, so in US, it's Lisa may probably know, each agency has their own funding programs. For example, the National Cancer Institute has, the centers, but then as I don't know of a large overarching program, but Lisa may know, DOD has their big programs, and then if you look at the NNI budget supplement document about the budget, so it's 20 billion spent over the years.
How Europe tries to build Europe with Horizon 2020, they want to have a spanning for all Europe available program. And I asked myself, that actually I feel does not exist really in the US.
Well,
It's
Europe
a different,
is, yeah.
it's a different, it's organized like Switzerland with cantons, and each one has his budget, each one has his initiatives.
Lisa, you want to? No.
I would say your observation is correct. We do it differently. But there are some activities that span across agencies and collaborative programs that are focused in certain areas. You know, so there are opportunities to discuss that, but we don't currently have a large-scale program like this for nanomedicine at this stage.
Okay, thank you. Thank you, Scott and Simon, Patrick. Again, thank you. So there are a couple of keywords that came up in the discussion so far. What is the regulatory science and the standards and US-EU cooperation. So the next two presenters are going to talk about this precise subject of standardization, which is very critical because even the definition of nanotechnology, which I don't want to bring up in this meeting, is then depending on how you define the nanomaterial, the methods that you have to use are going to be different. So if you need a number-based measurements versus up to 1 micron is considered as nano within FDA, beyond 100 nanometers.